Good afternoon. My name is Kiyoshi Woods. I'm a junior here at Rutgers University Camden. I'm a majoring in biology with minors in chemistry and psychology. Today, I'm here to talk about my project, which is observing DNA methylation changes associated with opioid use disorder. I'm going to be going through with a PowerPoint presentation rather than a typical poster presentation. I'll have my poster up as well in my booth. And if you have any questions regarding my results or anything else regarding the entirety of the project, feel free to stop in and chat. I'll be happy to answer any questions. So let's get started. So first, pain is the largest public healthcare crisis in the United States. Of the $635 billion are spent annually in costs associated for chronic pain patients. This affects more than 100 million patients, more than heart disease, cancer, and diabetes combined. Over 91 people die every day from opioid overdose, with almost half of these involving a prescription opioid. Specifically within Camden County, we are definitely afflicted with this. From 2010 to 2018, we have seen a 370% increase in fatalities from overdose, and not even within Camden County, but specifically within Camden City itself, we are the #1 in the county and one of the highest in the state. With between 2016 and 2018, we were #1 in the county for fatalities due to opioid overdose, with 35% of the total deaths coming from Camden. So in response to this, CORI was created. CORI is the Camden Opioid Research Initiative, which this was founded between the Coriell Institute for Medical Research, Cooper University Healthcare, and Cooper Medical School of Rowan University. This was tasked with the goal of defining genetic and non-genetic data to determine the utility of genetics in preventing new cases of opioid use disorder and overdose. This utilized samples from New Jersey and the Camden area specifically. So basically, they wanted to be able to take genomic and non-genomic data to be able to determine if genetically there are possibilities of susceptibility to opioid use disorder and any type of addiction. So in doing this, one of the things that we can look at is DNA methylation and epigenetics. Epigenetics is the study of how cells control gene activity without changing DNA sequence. And DNA methylation is the addition of a methyl group to cytosine followed by guanosine, or CpG dinucleotide. This does play a role in gene expression and epigenetics, and it's been used to estimate biological age, but it has other uses as well. So methylation can be altered by aging, sleep, and diet. It's bidirectional, so it can go more or less, and it can be changing throughout the purpose of your life. This can actually also be altered by opioid addiction and overdose. So CpG sites affected by methylation can be isolated to locate genes associated with this change. So the question we want to ask is, can this be used to find genes within the Camden population that put someone at risk of opioid addiction? That's the end goal, kind of where we want to see, and we're able to see where everything is. But first, our objective for today is, are there DNA methylation changes associated with opioid use disorder within the Camden community? So the methodology behind this is, first, we have to look at our groups. The sample groups we're going to look at are 4 different groups that were organized and pulled from the CORI database. CPMC is the samples from the Coriell Personalized Medicine Collaborative, and all apparently healthy samples. Opt-in was the Optimizing Pain Treatment New Jersey study. Individuals were all being treated for chronic pain and are not addicted to opioids, but are considered at risk. Goals is the Genomics of Opioid Addiction longitudinal study. All individuals are addicted to opioids and are medication-assisted therapy treatment at time of sample collection. And donor or biobank is unfortunately samples from individuals who passed away from opioid overdoses. In doing so, we just want to look a little bit at the demographics of each of these groups. We can see the ages here, where it kind of spreads out. And with sexes, we do overall have a roughly equal amount between female and male, but unfortunately, due to the nature of the study, we weren't able to fully get a complete half and half for each of these groups. So the first thing we want to do in this is to read and wrangle the metadata. Then we want to inspect the overall coverage of these CpGs to check for significance. So as you can see for here on the top right, where it says the PM0032, RRBS 5356, the ones are highlighted in blue, we wanted to be able to kind of look for, is there significant samples that have high amounts of CpGs, with CpG counts being on the left-hand side of the y-axis. So we wanted to filter for any CpGs with sufficient sequencing coverage, and we would remove anything that had a lower amount than we wanted to look at for. And so next, we wanted to confirm that there was an expected bimodal distribution for methylation data. So methylation is kind of like an all or nothing. So it's either left or right. So on the bottom right, that is going to be the expected bimodal distribution, where you can kind of see peaks on each of the sides of the extremes. If you see any types of issues resolving this, or you're seeing more peaks in the middle, it can be an issue with the bisulfite treatment that was utilized in being able to create this genomic data. Then we want to be able to run a PCA to look for clustering by cohort, and we wanted to see if it clustered by sex and age as well. Then, for the final part of this, we wanted to run a differential methylation testing. First, with this, you would fit the model to look at variation within groups to compare between groups. We want to make contrast. Then we would run a full differential methylation testing for comparisons between each experimental group and the control, and then we'd plot results using a volcano plot. All of this is fortunately being able to be undergoing through our studio. So all of this is code-based, and it's not really much wet lab-based. So the results of this, let's take a look. The PCA. On the left, we wanted to be able to take the PC1 and PC2, which are the best looks of the amount of genomic samples, and we were able to separate it by cohort, in color by cohort, and have shaped it by sex. So what we want to see here is we want to be able to see some type of clustering between these. So fortunately, you can see here, we do have a clustering with the biobank. This is most likely because biobank had a different type of sample, because due to the nature of the person passing away, we weren't able to get a blood sample, so a saliva sample was being utilized. So you may see differences with that, but you also can see that you can see the opt-in is most likely clustered by itself with a couple pieces throughout. The CPMC, which is the healthy samples, you're seeing a good distribution because you're getting a wide range of people between ages and all female. So you will see a good amount around that. Goals, on the other hand, you will see a little bit of clustering, but you will see it does kind of mix in with the opt-in, which is interesting. And then in terms of sex, female, male, you are seeing clustering between females, of course, for the CPMC. But in terms of the biobank, you are seeing a good amount of mix between the males and females. typically, if the sex chromosome, the Y chromosome, was utilized and looked at, it would drastically just change the course of the study, just due to the nature of the amount of methylation and CPGs that are present. You would, if you had any type of sex chromosomes that were being looked at, it would automatically separate by that. So being able to separate it by that chromosome, you can kind of see a better distribution. In terms of age, you can see a good amount of range between this, but you can't really see a good amount of clustering between this. So I decided to leave this out in the study because I found that because aging, methylation can change across age, it can show that things can be very weird in that aspect. So for the volcano plots, we have three different plots. We have the opt-in versus CPMC, goals versus CPMC, and biobank versus CPMC. These three plots were against the control to be able to look at the percent methylation difference across this. So we have 0.05%. I'm sorry about that. We have below a 0.05 for the p-value and the FDR value. And you can see here that in terms of each of these, so in terms of the opt-in, we were able to see a 20,419 CPGs that were located to be hypomethylated, so beneath the regular methylation control. And we can be able to see a 655 CPGs, which are hypermethylated, which is really nice to see. Goals, you can kind of see a little bit of an increase with 28,491 CPGs that were located in the hypermethylated and 751 in the hypermethylated. Each of these will also be looked at statistically significant ones. So the ones in red will be the ones that are statistically significant, not the ones in gray. In terms of the biobank, you do see a very weird thing with this. So instead of going up to 20 for the FDR, which is pretty much normal, it jumps almost 4 times that, with much more CPGs kind of being hypermethylated and hypermethylated. You're seeing anywhere from instead of a 20,000, you're seeing a 60,253 CPGs and 23,156 for the hypo and hyper respectively. This is very interesting. So in conclusion with this, in terms of methylation differences, we can determine that there is a large methylation difference within up and down regulated CPGs in the goals versus CPMC data and the opt-in versus CPMC data. Data supports the possibility that opioid use disorder does influence an individual's methylation in terms of both the hypo and the hypermethylation. However, there are areas of concern. While there is much larger difference with the biobank versus CPMC plot, errors may arise due to the type of sample that was taken from biobank samples, which is the saliva versus typical blood. Differences may also be true to the sex chromosomes associated with each sample. However, the Y chromosome was not utilized within this data due to epigenetic differences in sex chromosomes. Due to large variation in age among samples, methylation may also not be correctly observed due to the various impacts of diet, sleep, and age of methylation because those weren't looked at. More testing with less variation between these groups will be needed to conclude that the factor that does change methylation is indeed opioid use. So future work is we want to be able to see a couple things where this was kind of the next steps we took. We want to look at the heat maps for opt-in and goals, which I didn't really want to look at the biobank due to the aspect of so many things were different within that. So you can see that the p-value is relatively small for each of it, which is good. And you can see that in terms of the amount of methylation that's increasing, you can see a good amount that's above in the difference between the control and the case. Control being the control samples, case being the opt-in samples. So you can see in that differences, you are seeing a good amount of change that is increasing. Same for the goals as well. You are seeing a little bit more of a change, but at the same time, you are seeing a little bit of change between the samples and the differences. And then in upset plots, we want to be able to look at the differences in CPGs and the amounts that were found in each. So this was with the goals, opt-in, and biobank, and this is without the biobank. The one with the biobank, you can see as it decreases down all the way either to the association between opt-in and biobank, you can see anywhere that it cuts down from 28,000 to 5,000 CPGs that were found among each of these. However, with just the goals and opt-in, you can see that in terms of both of them, you can see that it cuts down to like the 16,000 just between the two, which is great. So we do hope to see a little bit more of these CPGs, and we can kind of start to be able to break it down where the locations are. So future work is we want to utilize data that is uniform across sex, age, and lifestyle if possible in terms of smoking, diet, and other factors to confirm if methylation change is indeed caused by opioid use. We want to look at specific differences between CPG sites in the goals and opt-in groups, look at CPG sites methylated within each other that are not seen in the other, and the control CPMC group. I would then want to compile a list of genes that are associated with the location of the methylated CPG sites, and then begin to research genes associated and test within a Drosophila melanogaster model, which you can see in a little bit better than in terms of just the human aspect, which I unfortunately don't have access to. So that concludes my presentation. Thank you so much. And if you have any more questions, please let me know, and have a great day.